Doctors had always told families the same thing. If your parents have type 2 diabetes, watch your diet, because it runs in the blood. But nobody could explain why some families carried this risk so much more heavily than others, even when their lifestyles looked identical. Then a team of geneticists went looking for the exact piece of DNA responsible, and what they found did not come from any human ancestor at all. It came from a species that vanished from the Earth tens of thousands of years ago. The discovery stunned the research community. Scientists had expected to find a modern human mutation, something that developed within our own species in the last few thousand years. Instead, they traced the riskiest version of the gene straight back to the Neanderthals, our closest extinct relatives, who disappeared around 40,000 years ago. Somehow, a piece of their DNA was still living inside millions of people today, quietly raising their risk of one of the most common diseases in the world. If you enjoy discoveries like this, stick around and subscribe, because I break down stories exactly like this one every week. Now let's get into what the scientists actually found. The story begins with a massive genetics study focused on Mexico and Latin America, regions where type 2 diabetes rates are unusually high. Researchers wanted to understand why. They collected DNA samples from thousands of people across Mexican and broader Latino populations and compared them against large databases of human genetic variation. They were searching for a specific signal, a stretch of DNA that showed up more often in people who had developed diabetes compared to those who had not. After scanning millions of genetic markers, one region stood out sharply. It sat on chromosome number 17 in a gene called SLC16A11. People who carried a particular version of this gene had a substantially higher risk of developing type 2 diabetes. This was not a small effect either. Carrying one copy of the risk version raised the odds meaningfully, and carrying two copies raised them even further. For a single gene, this was one of the strongest genetic risk factors for type 2 diabetes ever identified. But the real surprise came when researchers looked closer at the structure of this gene variant. It did not look like anything found in the genomes of chimpanzees, our closest living relatives. It did not match older versions of the human genome either. Instead, when scientists compared it against the genome of a Neanderthal, extracted from ancient bone fragments discovered in a cave in Siberia, the match was almost exact. The risky version of this diabetes gene was Neanderthal in origin. This meant that at some point in ancient history, a Neanderthal and an early modern human had a child together, and that child carried a piece of Neanderthal DNA forward into the human lineage. That piece of DNA has been passed down through hundreds of generations, all the way to people living today. To understand how this happened, you have to go back roughly 50 to 60,000 years, to a time when modern humans were migrating out of Africa and into regions of the world already inhabited by Neanderthals. These two groups were not strangers passing each other in the distance. They lived close enough, for long enough, that interbreeding happened repeatedly across different regions and different time periods. It was not a single isolated event. It happened again and again as modern humans spread into Europe and Asia. Every time this occurred, a small amount of Neanderthal DNA entered the modern human gene pool. Most of these fragments carried no real advantage or disadvantage and simply blended in. Some fragments turned out to be harmful and were slowly filtered out by natural selection over thousands of years. But a smaller number of fragments turned out to be useful, at least at the time, and those survived, spreading through populations as they expanded and multiplied. The diabetes-linked fragment fell into a strange middle category. It was not obviously harmful when it first appeared, and researchers believe it may have even offered a small benefit tens of thousands of years ago. But the world our ancestors lived in back then looked nothing like the world we live in now, and that difference matters enormously to how this story unfolds. Scientists studying the gene's function found that SLC16A11 plays a role in fat metabolism inside liver cells. It affects how the liver processes triglycerides, a type of fat that circulates in the blood. The Neanderthal version of this gene changes how efficiently liver cells handle these fats, and that change ends up disrupting the body's ability to properly regulate blood sugar over time. In an ancient environment of food scarcity, unpredictable hunting success, and long stretches between meals, a body that stored fat more efficiently and used it more conservatively may have actually been an advantage. It meant surviving through lean periods when food was hard to find. This idea is sometimes called the thrifty gene hypothesis. The theory suggests that certain metabolic traits that once helped humans survive famine and food scarcity become dangerous liabilities in a modern world filled with constant calorie access, processed food, and sedentary lifestyles. A gene that once protected someone from starving during a harsh winter thousands of years ago can, in a completely different environment today, quietly push someone toward insulin resistance and type 2 diabetes. What made this discovery even more interesting was where the gene ended up concentrated. Researchers found that the risk version of SLC16A11 is far more common in people with indigenous American ancestry than in people with primarily European or African ancestry. In some indigenous American and Mexican populations, close to half of all people carry at least one copy of the risk variant. In East Asian populations, the frequency is lower but still present. In African populations, where Neanderthal ancestry is minimal because early modern humans in Africa were less exposed to interbreeding with Neanderthals, the variant is nearly absent. This distribution pattern is not random. It connects directly to the story of human migration. The ancestors of indigenous American populations passed through a long chain of migration, moving out of Africa, through the Middle East, across Central Asia, into Siberia, and eventually across the ancient land bridge into the Americas. At various points along this journey, small groups broke away from larger populations to continue migrating further. Each time a smaller group split off, they carried only a portion of the genetic variation present in the larger group. Picture a large jar filled with hundreds of different colored marbles, representing the full range of human genetic variation available at that time, including the Neanderthal DNA that had already entered the population. Now imagine a small handful of marbles being scooped out to start a new, smaller population somewhere else. That handful will never contain the same balance of colors as the original jar. Some colors might be missing entirely. Others, purely by chance, might end up overrepresented in the new, smaller group. This is called the founder effect, and it happened repeatedly during the long migration into the Americas. Each time a population split and moved further, certain genetic variants became more concentrated purely by chance, simply because the founding group happened to carry more copies of them. The Neanderthal diabetes variant appears to have been one of the variants that, by chance, ended up carried more heavily by the small founding populations that eventually settled the Americas. Over thousands of years and countless generations, that starting frequency shaped the genetic makeup of an entire hemisphere's worth of descendants. This is not the only piece of Neanderthal or archaic human DNA that shaped modern health in unexpected ways. Scientists studying high-altitude populations in Tibet discovered a gene called EPAS1 that helps the body use oxygen more efficiently in thin mountain air. This gene did not come from Neanderthals. It came from an entirely different vanished human relative called the Denisovans, a group closely related to Neanderthals that lived across parts of Asia. Tibetans today carry this Denisovan-derived gene at strikingly high frequencies, and it appears to be the reason they can live and work at extreme elevations without the health complications that affect lowland populations who travel there. Other archaic gene fragments have been linked to immune system function, skin and hair traits, and even how some people respond to certain infections. Small pieces of Neanderthal DNA have been found to influence susceptibility to conditions ranging from depression to nicotine addiction to blood clotting disorders. None of this means Neanderthals were sick or unhealthy. It means that different genetic solutions helped different populations survive different environmental pressures. And those solutions did not disappear when Neanderthals themselves went extinct. They simply moved into us. On average, people with non-African ancestry carry somewhere between 1 and 2% Neanderthal DNA scattered across their genome. That might sound small, but spread across millions of people over thousands of generations, small percentages translate into real, measurable effects on modern disease patterns. No single person carries a large block of Neanderthal genome intact. Instead, tiny fragments survive here and there, buried within an otherwise fully modern human genome, quietly doing whatever job that particular stretch of DNA does. This changes how we should think about family history and disease risk. When a doctor says diabetes runs in your family, that phrase usually makes people picture a risk that started with their grandparents or great-grandparents. But some of the genetic risk being passed down is far older than that. It did not start with a relative anyone remembers. It started with an encounter between two different human species tens of thousands of years ago, long before written history, long before agriculture, long before anyone had a word for diabetes at all. It also reframes what type 2 diabetes actually is, at least partly. It is not simply a disease caused by poor modern habits, even though diet and activity levels clearly matter enormously. For some people, a piece of ancient inherited biology is working against them before they ever make a single lifestyle choice. Their body is running metabolic instructions optimized for a world of food scarcity while they are living in a world of constant abundance. The mismatch between ancient biology and modern environment is where a significant portion of the risk actually comes from. Researchers are now using this knowledge in practical ways. Understanding that certain populations carry a higher frequency of this Neanderthal-derived variant helps explain some of the disparities in diabetes rates across different ethnic groups, disparities that were previously blamed almost entirely on diet, healthcare access, or lifestyle differences. While those factors absolutely still matter and remain hugely important, they are not the entire story. Genetics loaded some populations with a higher baseline risk before any lifestyle factor even entered the picture. That does not excuse unequal healthcare access or the very real social factors behind health disparities. It simply adds another layer of explanation that had been completely invisible until modern genome sequencing made it possible to see. This discovery also raises a bigger question about what it actually means to be human. For a long time, Neanderthals were seen as a separate branch on the family tree, a related species that eventually died out and left nothing behind except bones and stone tools in museum displays. That picture is no longer accurate. Neanderthals did not simply vanish, they were absorbed. Their genes mixed into the population of modern humans expanding across the planet, and those genes have been quietly riding along inside us ever since, occasionally showing up in unexpected ways, like a metabolic gene that changes how your liver handles fat. In a strange sense, Neanderthals never fully went extinct. Their species disappeared, but fragments of their biology live on, scattered across billions of people, expressed differently depending on which fragments each person happened to inherit. Every time a doctor mentions family history of diabetes to a patient with indigenous American or Latino ancestry, there is a real chance that a small, specific echo of that ancient interbreeding event is part of the conversation, even if nobody in the room realizes it. Scientists are continuing to study other genetic risk factors for type 2 diabetes, and this Neanderthal-derived gene is only one piece of a much larger and more complicated puzzle. Diabetes risk comes from dozens, possibly hundreds, of different genetic variants working together, combined with diet, exercise, environment, and overall health. No single gene determines anyone's fate completely, but this particular discovery stands out because it connects a modern chronic disease directly back to an ancient chapter of human history that most people never think about. It also serves as a reminder of how deeply connected human history really is. The migrations that carried small groups of people across continents thousands of years ago still shape the health of their descendants today. The choices ancient humans made about where to live, who to have children with, and which direction to travel next were never really choices made with modern medicine in mind. Yet those decisions rippled forward through time, shaping which genetic variants became common in which populations, and ultimately shaping which diseases certain groups of people are more likely to face today. The next time someone mentions that diabetes runs in their family, there's more happening beneath the surface than a simple inherited habit or a shared diet passed down through generations. Somewhere in that inheritance, hidden inside a single gene on chromosome 17, might be a fragment of DNA that has been traveling through human bloodlines since long before recorded history began, carried forward from a species that no longer exists, but that never really left us behind. What the scientists uncovered was not just a genetic risk factor. It was a direct physical link between the disease affecting someone today and a vanished human species that lived tens of thousands of years ago. It is proof that extinction is not always as final as it seems, and that the past does not stay in the past. Sometimes it survives quietly inside our own DNA, still shaping our lives in ways we are only now beginning to understand.